All right, good afternoon. It's great to be here to tell you about what we do at Senti. Senti is a company in South San Francisco. We started in 2016. Since then, we've grown to about 50 people in South San Francisco and really focused on doing synthetic biology for next-generation cell and gene therapies. So let me tell you a bit about what we do and really what the core technology around. It really centers around this idea of building genetic circuits inside of cell and gene therapy products. So we envision a future where these products are dynamic, controllable, and increasingly programmable, so that you can think about cell and gene therapy products that can sense disease biomarkers, compute decisions, and then translate those into therapeutic functions. We've been working on this for quite a while and have actually demonstrated that these gene circuits really are quite applicable across multiple modalities. We can use them to regulate gene therapies, to create more targeted or controlled gene therapy products, and I'll show you some examples of that going forward. And certainly we can insert these into many of the cell types that people are working on here, stem cells, immune cells, et cetera, anywhere where you want to increase the underlying functionality of those modalities. So we're quite excited about the opportunity to take these gene circuits and actually apply them quite broadly. Internally, we're focused in oncology for our internal pipeline programs, but very interested to work with other groups who are interested in areas outside of oncology through partnering collaborations. Just briefly as a history for the company, we've been working on the field collectively as the executive team as well as founders for almost 2 decades, have published actually much of the leading work showing that you can use gene circuits to treat a variety of diseases with this sort of approach. Personally, I left MIT in 2016 to lead the company. I've got very excited about the opportunity to take synthetic biology and combine it with the cell and gene therapies that are making their way into the clinic now and trying to think about the future generation of those products. We've assembled a great team of people to lead the translational biology within the group. So Phil Lee, my co-founder and chief operating officer, really specializes in the technical operations and manufacturing side of the company. Kurt Herbert, who's here in the room, is formerly at Sangamo Therapeutics and now leading our business strategy efforts going forward. And finally, Gary Lee, our chief scientific officer, spent 13 years in the cell and gene therapy space working at Sangamo, has taken 5 cell and gene therapies into the clinic, so a lot of experience on translational and regulatory. We started this company really to really pull together many of the tools that have been developed in the field of synthetic biology over the last 10 or 15 years. So in addition to myself, we have IP in license from universities as well as co-founders that continue to support the development of these genetic circuits and switches from places like MIT, working closely with Jim Collins, Wilson Wong and Mo Khalil, both at Boston University, Martin Fussenegger at ETH Zurich, and Chris Voigt at MIT. From a company build perspective, we raised our Series A in February 2018, a group of great investors that have continued to support our vision, and we're looking forward to continuing to scale the company in the coming years. So today, I thought I'd briefly tell you about how we use these genetic circuits to build our own internal pipeline. I'm going to really highlight a couple parts of our internal programs focused on, number one, how do we use synthetic biology to try to overcome challenges in solid tumors? Can we engineer cells that can modify that tumor microenvironment in a controlled way? And we're actually doing this with multiple cell types. So one of the things that we think very much about at Senti is how do we use gene circuits to enable a variety of different cellular modalities and take advantage of their beneficial properties. So we're using MSCs, mesenchymal stem cells, as well as T cells for our therapeutic programs for solid tumors. And then the remaining time, I'll briefly highlight some of the work that we're doing on the gene therapy side of things. And it's an area that we're very excited about and actively interested to push forward as well. So I'll start with the first program, which is what we call Senti 101. It's an engineered MSC product that's meant to really modulate the tumor microenvironment. So as many of you, I'm sure, know that in cancer immunotherapy, we want to try to engage and recruit the immune system to help us tackle the challenge of cancer. In our opinion, the sort of one of the ideal ways to try to tackle this is to recruit not just one cell type, not just T cells, but try to recruit the entire endogenous immune system into the fight. And the challenge with that ultimately is that we need to come up with ways of doing combinations or really having a more holistic immune response. So we're going to do that using engineered MSCs. And so the general idea is we're using MSCs that can basically home into tumors based off of recognizing chemokine gradients. And what we did was to spend about 10 months systematically building and screening hundreds of different immunomodulatory combinations that we could put into these cells, and then subsequently characterizing them across a variety of disease models to identify those that have a really strong, potent effect locally in that tumor microenvironment. The reason why we're particularly excited about this approach in general is that we'd like to enable what we think about as sort of artificial pharmacology for these IO products. So with Senti 101, because these cells are actively homing to tumors, we can achieve high local concentrations of expression. These are expressed over a much longer period of time than you would get with recombinant proteins. And we're getting hundreds of fold higher expression of these molecules than in the serum. Compare this to delivering recombinant cytokines, for example, directly into the bloodstream where you get very short half-lives, minutes to hours, poor tissue penetration into tumors, et cetera. So those are the problems we're trying to overcome. So what we've done within the company is really systematically build a design-build test cycle for these gene circuits. So we've built about 250 different combinations of cytokines, chemokines, antibodies, et cetera, that we can then deliver into any cell type of interest. In this particular application, we're using the MSCs essentially as a carrier to express these locally within the tumor microenvironment. And so here's just an example of the variety of different payloads that we've built and optimized for expression in these cells, as well as in other cell types like T cells. A variety of immunostimulatory cytokines, chemokines that can recruit the immune cells into the tumor microenvironment, growth factors, co-stimulatory domains, and even antibodies. We've systematically built individual versions of these as well as pairwise and three-wise combinations because we think one of the benefits of a cell therapy in this case is the ability to deliver combinations into that tumor. And so as I mentioned, we've systematically screened these in a variety of tumor models, both warm, cold, and hot. This is one example in a very significantly checkpoint unresponsive tumor, B16F10, where we look at Senti 101, which expresses two different payloads, effector 1 and effector 2, and we can achieve long-term survival here in over 50% of the mice. We're getting, this combination therapy is indeed also better than effector 1 on its own or effector 2 on its own, and this is with a single dose delivery into the animals. We've also demonstrated that this is a very scalable platform for manufacturing. So the MSCs can be harvested from healthy donors and used in an allogeneic fashion. We've shown that we can actually expand these cells many, many generations, creating tens of thousands of doses from a single donor. We can freeze these cells down, expand, and really redeliver them. They're still functional in vivo. So quite excited about the eventual deployment of these types of cells into the clinic. So next steps for this program, we're on track for our upcoming pre-IND meeting. We're completing the IND-enabling studies and in GMP manufacturing currently, and targeting an IND with this program in the second half of 2020. Now, this is just one example of how we're using cells to really deliver a combination immunotherapy into patients. We can also think about delivering these sort of combinations of payloads through other modalities, including T cells. So one of the other things, the second program that we're developing, Senti 301, is really trying to insert similar combination payloads, but into T cells that actually have effector function, that are directed towards a cancer antigen. The one unique part that we're bringing to the table here on top of that is that we can regulate the production of these payloads. So we know that CAR-Ts, TCR, any T cell therapies have sort of complex PK/PD properties once you've delivered them to patients. And so for us, it's important to be able to have on or off switch regulation of these products. So we have multiple technologies that enable us to do this. ProDial is one example of this, where we can use FDA-approved drugs. Basically, these are protease inhibitors that can be targeted to up or down regulate gene expression inside of the cell. These are FDA-approved drugs, orally dosable, and we think quite generalizable between both cell therapies and gene therapies. This is just an example of this in action. This is in primary T cells. On the left here is showing that we can control CAR expression. In this case, the CAR is fused to a fluorescent protein. We can turn expression of that on about 28-fold with a small molecule drug. On the right-hand side of this graph, we can actually build both on and off switches of this system. So we can get, in terms of controlling payloads that are actually produced, up to 66-fold on in this particular case, as well as about 25, 26-fold off in this case as well. So we think this is quite exciting. We've picked our own favorite CAR-T targets to go after with this, really prioritizing ones that have been in the clinic before, demonstrating safety, some evidence of efficacy, but ones where we can really boost activity. We are quite interested in taking this module and porting it into other systems. And so for those of you in the crowd who are interested, where if you're developing other cell therapies, T cells, NKs, et cetera, and you want to boost the production of immune effectors from those systems for better persistence or activity, we'd be happy to chat about potential collaborations there. All right, so I briefly mentioned as a synthetic biology company, we're really focused on how do we show the utility of gene circuits quite broadly. Cell therapy is certainly one exciting use case, but we think gene therapy can also benefit from enhanced programmability and control. So there's really two major classes of products and technologies that we're developing here. On the left-hand side, number one, being able to regulate the expression of a transgene. So we think to be able to take gene therapy to indications where the therapeutic window might be tighter, be able to turn up or turn down the expression of that transgene on a dynamic or a regulated basis would be very powerful. Secondly, we want to be able to enhance the programmability of these therapies. So can we get cell type specific expression of the payload and also maintaining really high level production when it's actually needed. So these are the two technology pieces I'll show you in a bit. On the first one, I alluded to the idea that we have small molecule regulators that can be used to control on switches or off switches. In this particular case, we've encoded one of those off switch formats to regulate a luminescent payload just as a proof of concept in an AAV context. And as you can see here with both construct 1 and construct 2, at high levels of the drug, where we're basically shutting down expression of this transgene in the AAV. But when you basically dose titrate down the drug, you basically enable high-level expression to turn on. So this is one way in which you can essentially tune gene expression by dosing, in this case also an FDA-approved drug, into AAV transduced cells. Secondly, we've done a lot of work around engineering synthetic promoters, UTRs, and then ultimately logic gates to give us very enhanced specificity for targeting specific cell types of interest. So in this particular case, we've come up with a high throughput platform recently published a few months ago, where we can build large libraries of artificial promoters, tens of thousands, hundreds of thousands at a time, transduce these into cells of interest, and then using a variety of high throughput methods, basically pick out promoters that are active in the cell types of interest. In this particular experiment here, we've basically tested a library of these promoters in 2 different cell types. This is a breast cancer cell line, MB453 cells versus these MCF10A cells. And we actually have picked out promoters that give you selectivity for either one or the other. So on the left-hand side of this graph here, you can see in the blue, we get high-level expression, orders of magnitude higher level expression in the blue cells versus the red cells. And if you want the opposite behavior, we can also find promoters that give you that sort of activity. The second thing to really note here is that we're actually able to find promoters with different expression strengths. So if there's a need to really tune expression to a certain level, or if you're just trying to maximize expression, we think we have a great platform to try to enable that. Obviously, promoters are not the only control system that we have to regulate transgene expression. We can also do this using UTRs. And so we've done similar methods, building UTR libraries, doing similar types of screens, and then really combining this with an underlying machine learning model so that we can actually forward predict in the future what are the best UTRs for a given context. This is just one example comparing some of the synthetic UTRs that we've built to a DNA vaccine vector that's commonly used, where we can show increased expression, especially for this Neo UTR3 here. So one of the really unique things I think what Senti brings to the table is now if you have synthetic promoters or you have synthetic UTR sequences, that gives you some level of regulation over the expression of those transgenes. What if you want to get even better in terms of specificity? And so one of the great things that we've been able to bring to the table is this idea of logic gating. So this idea that you can build gene expression constructs where you can make the expression of that transgene conditional not just on one promoter, but 2 promoters. We call those AND gates. In this particular case, both promoter 1 and promoter 2 need to be active together in order to then trigger the expression of the output of interest. So here, in this particular case, we've done this in the context of a cancer application where we're trying to specifically target expression of really potent immuno payloads, things like IL-12 or checkpoint inhibitors, but do that only locally within the tumor microenvironment. So we went through this process of screening promoters, identified ones that were driven by certain cancer-associated transcription factors like c-MYC, and showed that we could actually put together a virus that basically could transduce cells and gets very specific expression of these payloads. And we've demonstrated in a variety of in vivo models that you can actually achieve anti-tumor efficacy using this sort of approach. So ultimately, we were quite excited about the opportunity of this to try to expand gene therapy beyond constitutive expression of a single transgene in limited tissues. If we want to think about applications where there's a narrow therapeutic window where you can't go too high or too low, or you want to be able to achieve cell type specific expression, these are some of the applications that are enabled by our tool set here. So in summary, we've come a long way. The company was Series A funded in the beginning of 2018. So in the last year and a half, we really achieved multiple things, going from a technology-focused company to building products. Senti 101, we nominated a development candidate at the end of 2018, have initiated CMC, clinical, and regulatory to move that into the clinic by the second half of next year. We have an in-house process development, quality, and manufacturing team that really oversee that whole process. For Senti 301, which is that solid tumor set of T-cell programs. We've initiated an R&D program and demonstrated really key proof of concept data there. We've also shown that we can actually build that pro-dial regulator system and apply it in both primary T cells as well as in gene therapy context, and also initiated a research collaboration with a large biopharma company. Really where we're going with this in 2020 is to take that initial program into the clinic. We're planning to initiate the phase 1 clinical study in ovarian cancer and then expand to other solid tumors. For Senti 301, we're on track to nominate our lead development candidate next year. And then sort of more broadly, probably one of the takeaways here is that we're really excited as a company to partner with other groups that are out there. Folks who have novel delivery methods, whether it's a virus, whether it's a cell type, or a specific disease focus. If we can help enable regulation, control, or dynamic expression of whatever therapeutic payloads you need, please come talk to us. We'd love to have that dialogue with you. So with that, like to end there, and happy to chat with you guys afterwards. Appreciate it.